IB Bio, Cell Structure and Function Part 9, is focused on the origin of life on Earth. The essential idea is that there is an unbroken chain of life, from the first cells on Earth to all cells and organisms alive today. Here is the outline of all the movies available for studying IB cell biology. Use this slide to reference the part of the topic that you need to review. This movie is focused here. In focusing on the origin of life on Earth, we can essentially return to cell theory, something that I presented in Cell Biology Part 1. Schleiden with plants and Schwann with animals specified that all living things were composed of cells, that a cell is the smallest unit of living things. Virchow insisted that all cells come from pre-existing cells, and that brings us to the subject of this movie, what might have been the origin of cells. Here's the first IB syllabus statement of this movie. State that cells can be only formed by division of pre-existing cells. State that the first cells must have arisen from non-living material. Explain briefly how this might have happened. Again, in the syllabus statement on the last slide, you heard Virchow's contribution to cell theory. All cells must come from pre-existing cells. In this micrograph, we can see a prokaryotic cell dividing through binary fission. In this diagram, we see many cells undergoing division. Here's another image of cells dividing, resulting in new cells. But at some point in the Earth's history, cells must have arisen from non-living material. Pasteur's experiments demonstrated that cells must come from other cells. He boiled nutrient broth and put it into 2 flasks. He used bent glass to prevent the entry of air and microorganisms into this flask. In this flask, he left it open. The flask that provided no access to microorganisms did not grow any cells, did not grow any bacteria, while those left open to the air did. All cells must come from pre-existing cells. Pasteur's experiments also provided evidence that the spontaneous generation of cells does not occur today on Earth. In returning to this same IB syllabus statement, let's focus on how the first cells must have arisen from non-living material. In the 1950s, various experiments were conducted, some of the best known by Stanley Miller and Harold Urey, in which small organic molecules, methane, ammonia, hydrogen gas, were placed into a closed glassware system. The solution was heated, evaporated, cooled, condensed, and sparked. After time, amino acids were found in the solution, evidence that the early Earth could have had conditions suitable to the formation of molecules that were important to forming the first cell. Here's a photograph of Stanley Miller standing next to his closed glassware apparatus, within which small organic molecules were formed, specifically amino acids. Now, the organic molecules formed in his experiment would really be the first step toward the generation of the first cell. The second step would be the formation of complex polymers. The third step would be the formation of a replicating molecule such as RNA. And the last step would be the formation of a membrane such that the internal chemistry of the first cell would vary greatly from the external chemistry surrounding the cell. Deep-sea hydrothermal vents are thought to be locations where the Earth's first cells appeared. Available inorganic molecules and heat from the vent would have been important in the forming of the organic molecules, the small organic molecules, not unlike what Stanley Miller got when he did his experiment in his closed glassware system. Secondly, after the formation of small organic molecules, then large organic polymers must form, and then a replicating molecule such as RNA, and then ultimately a membrane which encloses the unique chemistry within the cell. Volcanoes are also thought to be locations suitable to the formation of the first cells. Again, the abundance of inorganic molecules and the energy provided by the active volcano would have been important in forming the first small organic molecules, and then complex organic polymers, and then a replicating molecule such as RNA. As well, comets and meteorites may have delivered organic compounds to Earth. Amino acids have been found on meteorites that have arrived from space. Studies have also found that phospholipid molecules will spontaneously form membranes in a spherical shape when placed in water. Now, studies have found that phospholipid molecules will spontaneously form membranes in a spherical shape, as can be seen here. The formation of membranes might have been the last step after the formation of small organic molecules, then after the formation of complex organic polymers, and after the formation of a replicating molecule such as RNA. The membrane would have separated the internal chemistry of the cell from the external chemistry, and the internal and the external would vary greatly, and thus the first cell would have come to be. The earliest known cells are thought to be approximately 3.5 billion years old. These structures here, known as stromatolites found in Australia, are 3.5 billion years old, and they've been produced by cells. So the next IB syllabus statement for this movie is describe the evidence for the endosymbiont theory to explain the origin of eukaryotic cells. The endosymbiont theory applies to a time after cells had already formed. The endosymbiont theory explains the origin of eukaryotic cells, as the theory suggests that cell organelles, mitochondria, and chloroplasts evolved from formerly free-living prokaryotic cells that were engulfed by another prokaryotic cell but did not get digested. Instead, a mutualistic relationship occurred where the two cells together were more competitive, and this led to the origin of organelles such as the mitochondria and chloroplast. Here's a summary of what endosymbiosis is. It's a process by which unicellular organism engulfs but does not digest another cell, which lives within the host cell and ultimately becomes an organelle within the host cell, thus the origin of the eukaryotic cell. Mitochondria and chloroplasts were formerly free-living, small prokaryotic cells that were engulfed by a larger cell to become a more complex organism, the eukaryotic cell. In this diagram, you can see an original cell called a host cell. We'll ignore the idea of whether it was prokaryotic or eukaryotic to get to the fundamental idea of endosymbiosis. This bacterium is engulfed by this host cell, and you can see that process here, but the bacterium is not digested and instead becomes part of the larger cell, ultimately becoming the mitochondrion. The host cell and the bacterium together are more competitive. The origin of eukaryotic, heterotrophic, proto-animals has begun with this step of engulfing a bacterium that conducts respiration, and thus the evolution of the mitochondrion. Again, the mitochondrion is thought to be the descendant of a formerly free-living prokaryotic organism. In a second event, a larger host cell engulfs a photosynthetic bacterium, as you can see here, ultimately becoming the chloroplast. The alliance, then, is more competitive, and the origin of autotrophic, eukaryotic, protoplants has begun. The chloroplast is thought to be the descendant of a formerly free-living, photosynthetic, prokaryotic organism. So what's the evidence for the endosymbiont theory? Well, here it is. The first is that there's a similarity in size between the mitochondrion and the chloroplast and prokaryotic cells. The second is that mitochondria and chloroplasts have their own DNA. Check that out. And the base sequences in the DNA of mitochondria and chloroplast resemble the base sequences of prokaryotic cells. And mitochondrial DNA, chloroplast DNA are similar in structure to prokaryotic DNA, in that there's no histone protein around which the DNA is wrapped. And the protein synthesized by mitochondria and chloroplast resembles prokaryotic protein. The mitochondria and chloroplasts have their own ribosomes, and those ribosomes are similar in structure. They're small, just like prokaryotic ribosomes, the 70S ribosomes. And lastly, mitochondria and chloroplasts replicate using binary fission, just like prokaryotes. Here's an electron micrograph of a mitochondrion. The mitochondrion is now an organelle of eukaryotic cells that has its own DNA and ribosomes, similar to prokaryotic cells. The protein synthesized by the mitochondrion resembles prokaryotic protein. Mitochondria are thought to have descended from formerly free-living prokaryotic cells. The double membrane of the mitochondrion, which you can see the outer membrane and then the inner membrane, is probably a remnant from having been engulfed and surrounded by a second membrane. Here's an electron micrograph of a chloroplast. The chloroplast is now an organelle of eukaryotic cells that has its own DNA and ribosomes, both similar to prokaryotic cells. The protein synthesized by the chloroplast resembles prokaryotic protein. Chloroplasts are thought to have descended from formerly free-living prokaryotic cells. The double outer membrane of the chloroplast may be a remnant of having been engulfed and surrounded by a second membrane. In this diagram, we see a possible evolutionary tree for the three domains of eubacteria, archaeans, and eukarya. The common ancestor for all living things is here, while the last common ancestor of eubacteria and archaeans and eukarya is here. The last common ancestor of the archaeans and the eukarya is really here. Now, archaeans, while prokaryotic, they have DNA with histones, and it's thought that archaeans are more closely related to eukarya than eubacteria are to eukarya. Now, while we have prokaryotic cells here, there are some that perform aerobic respiration and some that can photosynthesize. Then here, one cell engulfs another to form a eukaryotic cell, a heterotroph. This is the origin of mitochondria. All eukarya have mitochondria. Later in time, along a different branch, a second endosymbiont event occurs where a photosynthetic prokaryotic cell is engulfed, and this is the origin of chloroplasts. This is the line leading to eukaryotic autotrophs, the photosynthesizers of the earth. The endosymbiont theory explains the origin of eukaryotic cells. A eukaryotic cell can be seen here with mitochondria. It's a fungal cell, thus there is a cell wall, but there are no chloroplasts. The evidence for endosymbiosis is substantial. Mitochondria and chloroplasts both have DNA. They have a single circular strand, just like prokaryotes today. The DNA of mitochondria and chloroplasts is not associated with histone protein, just like that of prokaryotes today. Mitochondria and chloroplasts can replicate, much like modern prokaryotic cells, binary fission. Mitochondria and chloroplasts have small 7ds ribosomes, just like prokaryotic cells. Mitochondria and chloroplasts are the same size as prokaryotic cells. Mitochondria and chloroplasts have double membranes. One more thing about the origin of life on Earth, and it's quite profound, is that the genetic code written in DNA bases is shared by all organisms on Earth. The DNA bases is transcribed into RNA, which in turn is translated into amino acid sequences called polypeptides. The translation of the code using triplets of RNA bases called codons to code for one amino acid is universal among all of life. The specific amino acid coded for by each triplet is universal. For example, all organisms everywhere translate UUU on the RNA as phenylalanine, the amino acid phenylalanine. GGC is translated to the amino acid glycine in the polypeptide chain. This is universal across all of life. This is the Codon Dictionary. Every triplet of RNA bases reading from the inside to the out codes for a specific amino acid. The system of genetic coding is universal, shared by all living organisms. The codons, the triplets of RNA bases, are translated similarly in all organisms on Earth. And that brings us to the end of IB Bio, Cell Structure and Function, Part 9. This is the last movie of the cell biology series.